Hello students, today we study IGCSE Biology, lecture number 17, Sexually Transmitted Infections. Our chapter is Sexual Reproduction in Human or Reproduction. Sexually transmitted infection. What are these infections? These infections are also known as STIs. There are many types of STIs: chlamydia, gonorrhea, genital herpes, hepatitis B, HIV, HPV, syphilis, trichomonias. These act in their own way and spread via sexual activities. Their symptoms and their effects are different. Sexually transmitted infection is an infection that is transmitted via body fluid. These fluid may be semen or saliva and through sexual contact between two persons. AIDS and HIV are our main topic that we study in our syllabus. The initial of AIDS stand for acquired immunodeficiency syndrome, pattern of symptoms associated with a particular disease. In AIDS, our total immune system is become weak. Every infection that we have act very badly on our immune system, and our antibodies which produce to treat the infection don't produce because the HIV infection or virus act on our antibodies. The virus that cause AIDS is the human immunodeficiency syndrome or human immunodeficiency virus HIV. After a person has been infected, years may pass before symptoms develop. This virus takes some time to reproduce and show some symptoms. So many people carry the virus yet not show any symptoms. They can still infect other people because virus is present in their body, and with the sexual contact with other, the virus transmitted in other person. It is not known for certain what proportion of HIV carrier will eventually develop 30 to 50% or more. What proportion of HIV virus can develop in AIDS or that transfer HIV virus into other human beings? HIV or AIDS is transmitted by syringes, used medical apparatus in pregnancy, mother to children or baby, blood transfusion, organ transplant, unprotected sex. HIV is not transmitted by food and utensils, mosquitoes, clothes, toilet seats, touch or kiss. It is none. HIV is transmitted by direct infection of the blood. Drug users who share needles contaminated with infective blood run a high risk of disease. For example, we have a couple or we have two-person, one is diabetic and have also HIV, like, but the other people is only diabetic. If the diabetic with HIV use a syringe and the diabetic which don't have HIV use the syringe of the first people, there is a chance that the diabetic patient who have not HIV have developed HIV in him. It can also be transmitted sexually both between men and women, especially between homosexual men who practice anal intercourse. You might be surprised why anal course or why intercourse between men and women, because the epithelium of anus is very thin, so it can disrupt easily and the virus can transmit it from one person to another with the help of body fluids, semen, etc. Prostitute who may have many sexual partners are at risk of being infected if they have sex without using condoms and are therefore potential source of HIV to the others. Hemophiliacs have also fallen victim to AIDS. Hemophiliacs are the person which have hemophilia. They have to inject themselves with a blood product that contain a clotting factor. The hemophiliac cannot produce their own blood clotting factor, so uh, they have are donated blood to produce or to get clotting factor. Before the risks are recognized, infected carriers sometimes donated blood which can used to produce the clotting factor. Babies born to HIV carrier may become infected with the HIV either in the uterus during the birth from the mother's milk. There are three ways that the baby can have HIV from his mother: in the uterus first, but during birth second, and from the mother's milk third. The rate of infection varies from 40% in part of Africa to 40 to 14% in the Europe. If the mother is given drug therapy during labor and the baby within 3 days, this method of transmission is reduced. There is no evidence to suggest that the disease can be passed on droplet by saliva or by normal everyday contact. When AIDS first appeared, there were no effective drugs. People living with HIV work with a healthcare provider to choose an HIV regime. They can use some drugs solely or they can use the combinations of the drug. Today, a range of drugs that can be separately or as a cocktail slows the progress of the disease. Researchers to find a vaccine and more effective drugs is ongoing. There is a range of blood tests designated to detect HIV. These tests do not detect the virus but do indicate whether the antibodies to the virus are in the blood or not. If the HIV antibodies are present, the person is said to HIV positive. The tests vary in their reliability and some are too expensive for the widespread of the use. The American Food and Drug Administration claims a 99.8% accuracy, but this figure is disputed. Like, share or subscribe if you like my video. Thanks.